Good morning. Welcome to our grand rounds. It is my pleasure to introduce our grand round speaker today, Dr. Ying-Hui Fu. Dr. Fu is a professor in the Department of Neurology here at UCSF and did her PhD work at Ohio State in molecular biology and biochemistry, and then went on to do a postdoc at Baylor, worked in industry for a while, and has since had faculty positions at Utah and here at UCSF. And her area of work is identifying genetic mutations associated with disorders of myelin and also genetics of sleep, and using model organisms to be able to understand the pathways, the mechanisms, which I think makes her work uniquely powerful. I've had the pleasure and great good fortune to be able to collaborate with Ying-Hui related to the sleep work that she has been involved in. And I can say it's only through that experience that I became aware of the depth, breadth, and reach of her work. The number of genes that she has discovered for the disorders is so large that I can't vaguely keep them in my head, and it wouldn't unduly delay her talk for me to even read them as a list, so I'm not going to bother doing that. But just a few highlights are that she discovered the unstable repeat mechanism, which is one of the ways that we understand the timing at which some genetic diseases start. She identified the genes for Fragile X syndrome and a whole lot of familial disorders that touch all of us here in psychiatry. And related to sleep, which is the topic of today's talk, she has found that there are single gene autosomal dominant mutations that have high influence, huge control over some fundamental things about sleep that we never would have thought would be under such control, like how much sleep we need and the timing at which we go to bed. But the reach of that work is much broader than simply those mechanisms. She's finding links to many illnesses, many elements of medicine that show how important sleep is and sleep-related circuitry is for our health more broadly. And for me, I think a really critical part of this is that her work in identifying the mechanisms by which this occurs gives us really a mechanistic toehold to be able to move from the Humpty Dumpty paradigm that we're stuck in, where we wait till people fall apart and then we try to put the pieces together, to prevention, to having a way to think about what causes these problems, what makes people predisposed to them so we can intervene. Just a couple of the areas that she's shown impact on are, like, she had discovered that the PER3 gene, a clock gene, is linked to seasonal affective disorder. She has shown that having the genes associated with natural short sleep actually prevent the expression of Alzheimer's disease in mice. She's done work in so many areas I can't even tell you, autism related work and depression predisposition related work. And I mentioned this because I know she doesn't possibly have time to tell you about all of these, but I'm hoping she's going to tell you about a few of these. And it's just, fantastic to have her here. There's so much all of us could learn from her. Really appreciate you coming, Ying-Hui, and please join me in welcoming her.
Okay. Thank you, Andrew, for such a generous introduction and for the opportunity to be here and talking to you about my favorite subject, sleep, right? Not just the sleep science, the physical activity of sleep happened to be one of my favorite activity. So let me make sure this works. Oh, okay. So in our group in the last 25 years or more, we have been trying to address some of these questions, including why should we care about get good night's sleep? And what is a good night's sleep anyway, right? How do we define good night's sleep? And also, how do we get it, right? How can we make sure that we can get good night's sleep? So these are some of the questions that we have been trying to address in our group. But before I start, I want to acknowledge two pioneers in the human sleep behavior basic science field, which is Chris Jones and Louis Potacek. In 1996, they started this whole field, and I joined them in 1997 on this journey. So in the last 25 years or more, we have worked on 4 different human sleep behaviors: advanced sleep phase, and delayed sleep phase, natural short sleeper, and natural long sleeper. So what are these behaviors? So here, for most of us, we usually get up 6 to 8 a.m. and go to bed about 10 p.m. to midnight. For the advanced sleep phase people, they usually get up much earlier and go to bed much earlier. And so their entire sleep wake schedule is shifted forward. So we call them advanced sleep phase people. For the delayed sleep phase people, they go to bed late and sometime in the early morning hours and get up late, sometime in the early afternoon hours. So their entire schedule is shifted backwards or delayed. So it's delayed sleep phase. For the natural short sleepers, they can go to bed about the same time with most of us, but they get up much earlier, or they get up about the same time with most people, but stay up much later. So they need fewer hours of sleep a day. And so they are natural short sleeper. It can be anything in between those two. And so for me, natural long sleepers are people who need longer hours of sleep a day for them to feel good or feel well rested. So these 4 different sleep behaviors can be divided into 2 groups. One group is all about sleep schedule, and sleep schedule is all about what is the best time for you to get up and what is the best time for you to go to bed that will be best for you. Over the last 25 years, we have identified more than a dozen mutations for human sleep schedule phenotype. And we now know that our sleep schedule is partly, not entirely, partly regulated by our circadian body clock. And the other 2 group falls into the group about sleep duration. And sleep duration is all about how many hours of sleep we need to get that will be best for our total well-being. This thing is so our sleep behaviors actually are genetically programmed. In other words, our genes have a lot to say about how we sleep, right? Now, even though our sleep is genetically programmed, still studying sleep is challenging because our sleep behavior is heavily modified or affected by many external factors, including like how much light you expose yourself to, especially in the evening hours, whether you use alarm clock to shock your body awake before your body is ready to wake up, or the beverages you drink, the medication you take, job, school, all these affect. And for many of us, grand submission also affect our sleep behavior, right? So if we look at our sleep behavior here with the sleep duration as example, it has this bell-shaped normal distribution curve. And most people fall into this middle range here, somewhere between 7 to 9.5 hours. But there are these people fall at the very end of the bell-shaped curve, and a small number of people fall at the tail end. And these are people who carry strong genetic mutation that make them have stronger behavior phenotype. And we're taking advantage of these people that carry strong genetic mutation and identify their genetic genes and mutations to help us understand human sleep behavior regulation. So I want to emphasize here that we are only study normal human sleep behaviors. We're not study sleep disorder or sleep problems, but I'm sure that if we can understand how normal human sleep behavior is regulated, in the future, we will be able to help with sleep problems. So today, what I'm going to do is I'm going to focus, just talk the sleep duration part of our research because of time. So whether we like it or not, most of us do need 8 to 8.5 hours sleep, but we only get 6.5 to 7 hours. So we drink a lot of coffee and tea. Outside there is coffee. If you need coffee, there's coffee there. And so we have been studying this group of people. We call them familial natural short sleeper. We call them familial because they carry genetic mutation. So the trait runs in the family. So we call them familial natural short sleepers. So these are people either lifelong or most of their lives sleep 4 to 6 hours a day. And they stay very healthy when they are not, stay very active when they are not sleeping, and also live a long, healthy life. So this is activity recording for one of these subjects. So this person on average goes to bed 1 a.m. and gets up 6 a.m. So on average, this person sleeps 5 hours a day. So when these short sleepers email me, oftentimes they will tell me that they identify themselves as both morning lark and night owl because they get up early and stay up late, both. So this is something very interesting they always say. So we reported the first mutation in 2009. And right after that, a lot of people came to us and told us that they identify themselves as natural short sleeper. So over the several years after that, we look at many of these natural short sleepers, and we realized that there is a list of features that are very common for these natural short sleepers. For example, almost all of them are very optimistic, and they are also very resilient to all kinds of stressful conditions, and they are very active. They are very active when they are not sleeping. And another thing is that a lot of them don't experience jet lag like we do, right? They can fly through many different time zones and adjust to the new time zone very quickly. And some of them have very high pain threshold. They go to dentist or they have procedure they don't need painkiller like we do. And also some of them have extraordinary memory. And the best one so far I know is one person can speak 13 languages fluently. Right? And so, and another interesting thing is that they also seem to live a long, healthy life. Almost all of them. So the first part of my talk, I want to address this question. I want to talk about why. Why should we care about get good night's sleep, right? Why is it important? Because some people think sleep is just a waste of time. So why should we care, right? So before I talk about that, let's just think about aging for a minute. So based on public database, that average human lifespan extend about 2 years every decade. So right now, the average human lifespan for men is 80 years old, for women is 85. And so by the time year 2050 comes, for the average human lifespan will be approaching 90 years old. And by the time 2100, it will be 100 years old. So this is great. We can all live longer and longer. However, unless something change and change quickly, we'll always spend about a third to a quarter of our life in a state fighting against diseases. And that's this blue part of this figure. Now, ideally, what we want to be able to do is we want to be able to live a long life, but spend most of our life in a state that's healthy, and only the last little bit that we need to manage disease. And that's much better. So in other words, we want to be able to reach healthy longevity. And I'm going to show you some evidence that indicate good sleep actually plays a key role in helping us reach the goal of healthy longevity. So in the last 40 to 50 years, I would say, there are a lot of reports. If you look into the PubMed, there are a lot of publications about epidemiology evidence suggesting that if we subject ourselves to long-term sleep disruption or deprivation, we have much higher chance to get all kinds of diseases ranging from high blood pressure, high cholesterol, all the way to cancer and all kinds of diseases. And here is just some examples I listed here. So when we started to look at this short sleeper for a number of years, it was very curious and very puzzling to us, because on one hand, we knew that if we subject ourselves to sleep deprivation or disruption on a long-term basis, we have much higher chance to get all kinds of diseases. On the other hand, we have this group of short sleepers. They don't need much sleep, and yet they can live a long, healthy life. So after we thought about this over the years, several years, we realized that actually there is a simple way to explain this. So if we understand that our bodies actually is like a machine that never stop working, right? During the day when we awake, there is a whole list of functions our body needs to do. We need to be able to eat, talk. I need to be able to talk. You have to be able to listen to me. We have to be able to interact with people and working and all kinds of, or exercising, right? So that we can live a normal, healthy life. But in the night, when we go to bed, our bodies don't just lay there and stop working, right? Our bodies continue to work, but with another whole set of different functions, including remove toxins or dead cells, repair damages that we acquire during waking hours, generate more energies for the next day, and memory consolidation, right? And all these are very important functions that our bodies need to do when we are sleeping. So for me, a regular sleeper, for my body to complete all these important functions so that I can stay healthy, it requires 8 hours, right? And let's say that it can reach to a level of 95 or 90% completion of all these tests. But for the short sleepers, it only need 4 to 6 hours. And they probably can reach even closer to 100% completion than my body can. And that's why they can sleep less and live a long, healthy life, right? In other words, their sleep quality and efficiency is better. And this good sleep quality and efficiency then offer them protection against many diseases. So we decided to test this idea. And we just totally randomly chose Alzheimer's to prove this concept. Now, when I decided to do this study, I went and talked to many Alzheimer's people and realized that among Alzheimer's people, still there's some disagreement about whether A beta is more important or tau is more important. So we decided to test on both mouse models. So here what we did was that we crossed our short sleep mice to A beta mouse model called 5XFAD here, or the tau tango mouse model PS19, and see if we can see any change. So the first mutation we looked at is DAC2 was the first mutation we identified for short sleeper. So we chose DAC2 to start with. Now we found DAC2 mutation in somewhere around 2004, 2005. So that's way before CRISPR time. So the mouse model made back then were all human back transgenic mouse models. So here the control is the wild type DAC2 back transgenic mice crossed with 5XFAD, and you can see there are a lot of A beta plaque here at 6 months old. Now, DAC2 mutant crossed with 5XFAD, there's significantly less A beta plaque accumulation. So at that point, we thought, well, great, our hypothesis is probably right. However, there's some chance that this observation is specific to DAC2 gene and has nothing to do with sleep. So to confirm that, we then chose the second mutation here, MPSR1, and we crossed MPSR1 to 5XFAD. Now, MPSR1 mutant is a CRISPR mice, so the control is 5XFAD. So here in 5XFAD, you can still see a lot of plaques, and MPSR1 crossed with 5XFAD, significantly less plaque accumulation. So these are example slides. Now, of course, we deal with many mice and many slices, and the quantitative data is here. Here you see the gray color is the DAC2 wild type control, and the blue is DAC2 mutant. So both male and female, and both hippocampus and cortex, they show that DAC2 mutant shows significantly less A beta accumulation than the wild type control. But for the MPSR1 here, the mutant is red and control is black. We only saw significant change in the female, but not male. And we think that there are two reasons contributing to this. One is that 5XFAD mice actually is well established that female have much more robust phenotype. And the second reason is that MPSR1 is a CRISPR mouse model. And we know that CRISPR mouse model is much weaker. Their phenotype is much weaker when you are trying to recapitulate human conditions. These two reasons together, that we think that that's why we only saw significance in female. So for tau tango here, the DAC2 mutant crossed with DAC2 wild type crossed with PS19 still has a lot of tau accumulation compared to the PS19 control. And so here green color indicates the tau tango accumulation. Now DAC2 mutant crossed with PS19 shows significantly less tau accumulation. And the MPSR1 crossed with PS19 has a significantly less green color tau accumulation than the PS19. So here with the tau, we decided to use a third mutation to confirm this. And this one, we chose the ADRB1. Here, if you see ADRB1 crossed with PS19 shows significantly less tau accumulation than the PS19 alone. So with 3 different short sleep mutations, when we cross them to tau mouse model, they all show the same thing. They show significantly reduced tau accumulation. We also wonder whether the short sleep mutation can help with other neurodegenerative problems, for instance, synaptic deterioration and astrogliosis. And we found that indeed the short sleep mutation can prevent the synaptic deterioration and astrogliosis problem for the degeneration mouse models. So for the rest of this story, I'm going to focus on tau because tau is more interesting. So before our study, David Holtzman at WashU, his lab showed that mice during sleep can clear out the A beta and tau through their CSF, cerebral spinal fluid. So we decided to look into the CSF tau clearance to see if our mutant mice actually have higher tau clearance, and that can help us explain why we don't see tau accumulation in these mouse models. Interestingly, when we look at the DAC2, there's no difference between mutant and wild type. But for the MPSR1, not only we didn't see increased tau clearance, we saw decreased tau clearance. This was particularly intriguing because from our study on the MPSR1, we knew that MPSR1 mutant neurons are more active. More active neurons theoretically should produce higher extracellular tau. However, here we see the mutant have less tau accumulation and less tau clearance. So we decided to confirm that these mutant mice do indeed have higher extracellular tau. So what we did was that we isolate synaptosome from both PS19 and and MPSR1 crossed with PS19 and look at their extracellular tau level and confirm that these MPSR1 mutant mice do indeed produce higher extracellular tau. So they have higher extracellular tau, less tau accumulation, and less tau clearance through CSF, suggesting that there are other mechanisms involved in trying helping clear out all these higher extracellular tau. So we decided to look at the tau story a little bit further and decided to look at one region of the brain, which is locus coeruleus. We decided on this region because locus coeruleus is one of the earliest regions that you can see tau accumulation in both human and mice. So we thought that we would start with this region. And here you can see that in the PS19, there's at 8-month-old, there are a lot of tau accumulation. But for the ADRB1 cross with PS19, there's significantly less tau accumulation. Now, in fact, as early as 3-month-old, we already could see that there's a significantly less tau accumulation in the mutant mice. Now, this was, again, very interesting for us because from our study in the ADRB1 gene, we knew that ADRB1 is not expressed in locus coeruleus. So ADRB1 is not expressed in this region. However, the mutant can prevent tau accumulation in this region. So the most logical explanation for this will be that there are ADRB1 neurons in other parts of the brain that can project to LC, and these neurons projecting to LC are the ones that are responsible for preventing tau accumulation. So we went to look for where are these ADRB1 neurons that project to LC, and we looked at all brain and found one region that actually ADRB1 neurons in the central amygdala actually project to LC. So here is ADRB1 in the central amygdala co-localized with the marker here. And if you look into the locus coeruleus region, you can see blue, the green color projecting into the locus coeruleus, indicating that ADRB1 neurons in the central amygdala can project to LC. But this still doesn't tell us whether the mutant ADRB1 neuron can help prevent tau accumulation. So to address that, we decided to seed human tau fiber into mouse brain to see if we can confirm that we can see the difference. So this slide is just showing you that when we seeded human tau fiber, how much tau spreading or accumulation is time and dosage dependent. The longer you wait, after you seed, the longer you wait, the more you see it has more spreading. So here we seeded human tau fiber. Sorry. So here we seeded human tau fiber into the central amygdala of PS19 and PS19 crossed with ADRB1 mice. And we waited for 30 days and look at the central amygdala. And we saw that there are a lot of tau accumulation in central amygdala, indicating that the seeding experiment worked, right? The seeding worked. But if we look at this locus coeruleus region, there are tau accumulation in the PS19 mice, but there's significantly less tau accumulation in the ADRB1 cross with PS19 mice. So this is ipsilateral part of the brain. For the contralateral part of the brain, both control and the mutant have very little tau accumulation. So this data confirm that ADRB1 mutant neuron in the central amygdala somehow can prevent the tau accumulation in the locus coeruleus region. So those are all the data I want to show you today, but I think that we have evidence to suggest that this short sleeper mutation somehow can offer protection for these mice and presumably human from getting Alzheimer-like diseases. And we believe that this is going to be true for many other diseases as well. We just don't have time to check them one at a time. But so all this data actually raise a very important question, and I think Andrew also mentioned it, is that whether we should start think about using good sleep as a tool to help us delay in preventing diseases. Now, Alzheimer is a good model for degeneration. So we are also curious about whether sleep can help with developmental problems. So we decided to use autism spectrum disorder to test this. So the wild type mice you see here, that if you put a stranger mouse here in the white dot into a home cage mouse, what happens? They will start interact with each other, sniff each other, and get acquainted with each other. Now, for the autism mouse, here with the Sheng3, we use Sheng3 mouse model. Here, you will see the autism mouse will be try to avoid the stranger mouse here. This one is the stranger mouse here. Stranger mouse, anyway, you'll see. Now, if we cross our short sleep mice to the autism, you will see that almost completely reverse this social interaction defect of autism mouse model. You can see the here on the right side, you see this the double mutation don't have any problem, but the middle is autism. You see the mouse try to avoid that. So when we cross the short sleep mice into the autism mice, you can see that almost completely reverse this social interaction defect problem in the autism mouse models. So these are our preliminary data, and we're trying to understand how sleep can help with autism phenotype. So next part of the talk, I want to tell you some of the stuff that we have done recently, trying to address what is good night sleep, how is sleep regulated. Now, if we take a moment and imagine this beautiful picture or a big puzzle is sleep duration, quality, and efficiency regulatory mechanism. Of course, we don't know what it looks like, but just assume that this is the beautiful picture that we're trying to understand, a puzzle we try to put together. In 2009, we identified first piece of the puzzle that we can put down, which is a DEC2 gene. In 2019, we found 2 more pieces to put down. And more recently, we found a few more pieces. The problem is that all these different pieces seems to be not related to each other. There are genes involved in different pathways, right? So now the biggest question is, how do we go from there to here, right? How do we find these pictures? So the simplest way to think about this will be we use each one of these pieces to start build around it, and hopefully someday they all merge together and give us a picture of what this should look like. Right? Now, in the last 15 years, actually, there are several labs published many papers on both wake promoting and sleep promoting pathways or circuits. One issue with all this study is that they use wild type mice and destroy certain parts of the brain or neurons in certain parts of the brain and look for sleep-wake behavior change. Now, if you understand how complex sleep-wake regulatory mechanism, circuits, and molecular mechanism is, it's not too hard to see how if you destroy certain parts of the brain, you can offset the balance of the circuits, and therefore you can see a sleep-wake behavior change, right? And I think that's the reason why almost all disease of the brain, the patients also have sleep problems. So we thought that it would not be the wisest decision for us to base our investigation on results obtained from those that approach. So instead, what we should do is we should use the genes that we identified in human that we know 100% sure that they are related to human sleep-wake regulation or sleep-wake behavior, and use these genes to help guide us to find the important brain regions for sleep-wake regulation. So what we have been doing is we use the gene we identify in human, make mouse model, and use the mouse model to help us identify important brain regions for sleep-wake regulation. When we find the important region, we then will go on to look at the circuit, the cell type specificity, and the molecular mechanism. Now, of course, our goal is hopefully someday we can come back, help people, not just with sleep problem, but also everybody get good night's sleep every day. So today, what I'm going to do is I'm going to show you two examples of what we have done recently in helping us understand the sleep-wake regulatory mechanism. So the first example I want to show you is on the gene called ADRB1. ADRB1 encodes, hold on. ADRB1 encodes adrenergic receptor beta 1. And the mutation was identified for this family. The mutation carriers in this family sleep on average 132 minutes shorter than non-mutation carriers in this family. So for ADRB1, one thing we decided to do was that we look at ADRB1 expression pattern in the whole brain. And we look at all the literature and references, everything about sleep-wake regulation in the brain region. We combine the two lists and come up with more than 10 brain regions that we think is worth for us to go after. And we then systematically silence ADRB1 neurons one at a time in all these regions to look for sleep-wake regulation change. And out of all these regions, we found one region, which is in the tegmental reticular nucleus region. It's all around here, so we call it peri-TRN. Only in this region, if we silence ADRB1 neurons, we saw a significant sleep behavior change. Now, the first set of experiment we did was by infrared video recording. For the mammalian system, EEG, EMG is gold standard. So we had to repeat this with the EEG and EMG. And we used a very tiny amount of tetanus toxin to silence this neuron. And we still saw significant change in wake and sleep, indicating that these neurons do play a role in sleep-wake regulation. Now, when we look at the EEG data, we found that the mice that we silenced the neuron, they show significantly reduced wake bound number and reduced non-REM bound number, and significantly increased wake bound length and no change on non-REM bound length. And there's no change on REM sleep. So the only way we could get this data is that if we look at this, all the possible sleep-wake state transition, the only way we could get this data is if this mice stay awake longer and they don't transition from wake to non-REM. So you have a longer wake bound length and reduce wake bound number and reduce non-REM bound number and no change on non-REM bound length. So altogether, this actually suggests these neurons also play a role in wake maintenance. And I'm going to show you some evidence on that. So John Webb was one of my graduate students who did this part of the project. So one of the questions he asked was, are these neurons active during wakefulness or sleep? So he first used this photophotometry method to look at the entire ADRB1 neuron population in this region. And with this method, he found that these neurons are mostly active during wakefulness and REM sleep and quiet during non-REM. He then used, and this system is a microscope. You can look at one neuron at a time. And with that single neuron, he still saw they are more active during wakefulness and REM sleep. So it's all consistent that these neurons are wake and REM active, but non-REM quiet. He then wanted to see if he activated this neuron, could he then change their sleep-wake state, right? Is it strong enough to change their sleep-wake state? So he first used DRESS to activate this. DRESS is a method you can activate ADRB1 neurons specifically, and by giving them this compound called CNO. So he first did this experiment at the ZT1. So ZT0 is when we turn the light on. And mice are nocturnal. So when you turn the light on, is when they just go into rest phase. So ZT0 and ZT1 is when their sleep pressure is highest. They're just going into their rest phase. So when he did this experiment at the ZT1, he saw that if he activated this neuron, he saw significant increased wakefulness and decreased non-REM and REM sleep, indicating that these neurons are wake promoting. He then repeated this experiment at the ZT12. So ZT12 is when we turn the light off. It's at the end of their rest phase. They have lowest sleep pressure. They spend about 75 to 80% time awake. Even when they are mostly awake, if he activated this neuron, he still saw significantly increased wakefulness and decreased non-REM and REM sleep. So this was, again, support the idea that these neurons also have a role in wake maintenance. So he also used optogenetic. Optogenetic is more acute than DRESS. So here the blue bars indicate when he shined the light on these neurons. When he shined the light on the neuron to activate them, you see the significant increased wakefulness in blue here and decreased non-REM in purple here. The interesting thing here is that 3 minutes after he stimulated this neuron, the mice have much higher chance to be awake than 3 minutes before he activated this neuron. Now, 3 minutes in optogenetic is a long time. So this data also suggests that these neurons also play a role in wake maintenance. He then wondered, where do these neurons project to in the downstream pathway, in the entire pathway? Where are these downstream neurons that this neuron go to exert the sleep-wake regulatory mechanism? So he used two different methods. One is called MAPC, which is a quite involved procedure, and the other one is synaptophysing. With these two methods, he found that there are two regions that these neurons project to, two regions. One is anterior lateral hypothalamus, and the other one is posterior parafacial zone. And these two groups of neurons that are projecting from peri-TRN to these two regions are mostly distinctive because when he did the ritual B tracing experiment and inject green dye to lateral hypothalamus, red dye to parafacial zone, he saw very little red and green overlap in the cell body, indicating that these neurons projecting to these two regions are mostly distinctive group of neurons. Now, when he stimulated the end of the projection at the lateral hypothalamus, he saw a very similar thing that he saw in the cell body, increased wakefulness in blue and decreased non-REM in purple. Now, if he stimulated the end of projection of the parafacial zone, he saw increased wakefulness and non-REM, but not as dramatic as the lateral hypothalamus. Now, interestingly, he also saw increased REM sleep in this parafacial zone. Now, mice have very little REM sleep, so this is a significant increase in REM sleep. So projecting to these two regions, it has they have different functions. So from our study of ADRB1 neurons, I only show you peri-TRN region. We have done this for many other regions. From our study, we now know that even the same neuron, ADRB1, at the different parts of the brain actually play different role in sleep-wake regulation. Now, even in the same region, I just showed you in the peri-TRN, there are subgroup of the neurons can project to different regions and they do different things. So this all support the idea of how complex the sleep-wake regulation mechanism is. Now, the second example I want to show you is on the gene called NPSR1. And NPSR1 encodes the receptor for the neuropeptide NPS. And in this family, the mutation carriers on average sleep 180 minutes shorter than the non-mutation carriers. So we're currently wrapping up the study for NPSR1, like the one I just told you, ADRB1. We use the same approach to go in, look at many different parts of the brain to see if we could identify brain regions for sleep-wake regulation. And we have a few more very soon here. But for this one, we decided to also look at NPS because NPS and NPSR1, they are very interesting. They have this one-on-one relationship. The only agonist with the only receptor. And NPSR1 is widely expressed in many different parts of the brain, but NPS is only expressed in a small number of nuclei. And NPS is recognized as a wake-promoting neuropeptide. So we thought that we would also look at the NPS in addition to MPSR1. So with our MPS Cree mice crossed with reporter mice, we found that MPS is mainly expressed in these two regions, parabrachial nucleus and locus coeruleus, or around locus coeruleus, peri-LC. So these are consistent with previous publication. But in addition to these two regions, we also saw MPS neuron in the region called central gray of the palm. So we decided to look at MPS neuron in these three regions to see how they participate in sleep-wake regulation. So for the PB region, MPS neuron, if we silence this neuron, we saw significantly decreased wakefulness and increased non-REM and REM sleep. If we activated this neuron, we saw significantly increased wakefulness and decreased non-REM and REM sleep. So altogether, this would suggest that MPS neurons in these regions are wake-promoting neurons. Now, because MPSR1 is the only receptor for this peptide, so we thought that if we block this receptor, we should see the cancellation of this phenotype that we see here, the result we see here. So that's what we did. So this panel is just the same with what I showed you in the last slide. Here we repeated the DRESS experiment, but now we gave them the very specific antagonist of MPSR1 called SHA68. Now, when we did that, we saw the cancellation of the wakefulness result, almost complete cancellation of non-REM. But for the REM, there's still significant decreased REM. So this was interesting for us. We needed to confirm this. So we decided to cross MPS Cree to MPSR1 knockout mice. So now there's no MPSR1 in this mice, and repeated this experiment. And again, we saw the cancellation of wakefulness and non-REM, and REM still significant decrease. So all this will suggest that there are actually two pathways for MPS neurons in the PV region. One pathway is MPSR1 dependent, and that's for wake promoting. And the other pathway is MPSR1 independent, and this independent pathway is involved in REM sleep regulation. Now, for the peri-LC region, the MPS neuron, whether we silenced them or activated them, we saw no change at all, indicating that MPS neuron in this region actually don't play a role in sleep-wake regulation. For the CGPN region, if we silence this neuron, we saw significantly increased wakefulness and decreased non-REM and REM sleep. So this result would suggest that these neurons are actually sleep promoting. So this is interesting because MPS is thought to be a wake promoting neuropeptide, but here we see these MPS neurons are sleep promoting neurons. So we wanted to confirm this. So we used Cas-Rx to silence or knock down the MPS in these neurons and saw similar result that increased wakefulness and decreased sleep. And if we activate this neuron, we saw significantly decreased wakefulness and increased sleep. So altogether, it's consistent that MPS neuron in these regions are sleep promoting. So here with MPS neuron as example, I showed you three regions. One region they are wake promoting, one region they are sleep promoting, and one region they have no function, right? So this all is consistent with how complex the sleep-wake circuits or mechanism is. Now, what about their activity? We found that in the PB and peri-LC region, the MPS neurons are wake and REM sleep active here. I think pink is REM sleep and yellow is wake. So you can see both PB and peri-LC, these neurons are active during wake and REM sleep. So again, this is interesting because peri-LC region, MPS neurons don't seem to play a role in sleep-wake regulation. However, their activity still track with sleep-wake state, right? So interesting. Now for the CGPN region, MPS neurons are only active during wakefulness. You can see that they're only active in the yellow, which is wakefulness. So these neurons are sleep promoting, but they are active during wakefulness. So what I've shown you today is I've shown you three regions that we have identified with our own gene that are very important for sleep-wake regulation. And as I say earlier, that we are also doing the similar study for NPSR1 with the one I showed you, ADRB1. And very soon we will have a few more regions to put down in this short video here. So let me look at the time. Okay. So I think last few minutes, I want to just very quickly touch base on sleep and mental health. I thought for this talk, I should at least. So I think for this audience, I don't have to say anything more than resilience is very important for our mental health, right? It's directly connected to many mental health problems. So now, if you remember earlier, I say that these short sleepers that we're studying, one interesting feature that they all share is that they are very resilient to stressful conditions. So we thought that maybe we could look into our mouse model, see if we can use our mouse model to study resilience. Because right now, what scientists do for people study resilience is that they take wild-type mice and they subject them to stressful conditions and divide them into two groups, right? One group is more sensitive and one group is less sensitive to stress. And then compare the gene expression or circuit change between these two groups. There's really no good mouse model for resilience. So we wonder whether we could come up with one. So the first thing we had to do was to confirm that these short sleepers do indeed have a higher resilience than the non-mutation carriers, right? So this is a set of data obtained by Liza Ashbrook, a sleep clinician in neurology. So here, this column is for the short sleepers, and this column is for the unaffected family members. And this is their age, and there's a gender. This thing is difficult to maneuver. I'm not very good at this. But anyway, if you look at the sleep duration, the short sleepers sleep 5.6 hours versus 7.7 hours unaffected family members. Now, if we look at how sleepy they are, the short sleepers are 4.4 versus 7.2. So even though they sleep 2 hours or more shorter, they are less sleepy than the unaffected family members. Now, if we look at their resilience based on this score, they are the short sleepers score 36.5 versus 30.3, right? So this will confirm that these people, short sleepers, are more resilient than non-mutation people, right? So we decided to use two methods to test our mouse models. The first one is this chronic restraint stress methods. So we put mouse into a 50 ml test tube for 4 hours a day and repeat this 14 days. And then after that, we test their resilience. The second method we used is called chronic social defeat stress test. Here you subject mouse to a bully mouse just for a few minutes a day, like just a few minutes, and you repeat this 14 days. And then after 14 days, you see how resilient they are. So I'm just going to show you the summary of our preliminary data. These are not published. So I want to show you this very interesting data. So here, if we look at the wild-type mice, after they were subjected to this chronic social defeat stress test, subjected to bully, they lost 75% of their resilience. They only retained 25% of their resilience compared to before we subject them to the stressful conditions. For the short sleep mice, they still retain about half of their resilience compared to before the stressful conditions. Now, this chronic restraint method, it looks terrible, but actually it's much more milder. You can see that the wild-type mice, after 14 days, still retain 94% of the resilience. And our short sleep mice actually slightly increased resilience than before they were subject to this. So overall, this will confirm that our mouse model, we can use it as a mouse model to study resilience. And we're going to try to see if we can use them and understand the molecular mechanism and circuitry for resilience. So I want to thank my longtime collaborator, Louis Potashek, and my collaborator, Andrew Crystal and Liza Ashbrook, and also all the people in the lab actually doing all the hard work. I'm only talking, but they are the people who actually do all the hard work. Thank you.
Gave a long list of reasons why it's good to be one of these people with short sleep. Everybody no downside?
So, well, you know, you never can say 100% sure, right? But we have yet to see what's the downside. Yeah. I think there are probably, but they don't tell me. Maybe that's the right answer.
You got a couple chairs here that you could ask.
Yeah.
I'm certain we have. I can think of one that is absolutely has to be a familiar short sleeper, close colleague.
Oh, I know one. He won't participate. He won't. He's like back off.
So one, and this could be totally wrong, but we're in the right audience to ask about this. I wanted to ask you about sort of the underlying model, but before I just had a thought about, so when you start showing psychiatric disorders, right? I mean, there's a huge range of psychiatric conditions where sleep is disrupted, right? You think about pretty much all our disorders that fall into sort of cognition, both kind of social and general cognition, all of our mood and anxiety disorders, all have prominent sleep problems. I wonder if people, so my impression just thinking about it is that at least in clinic, I don't think I've seen that with at least the habit disorders that I've been involved with clinically. So Tourette syndrome, unless there's comorbid ADHD, don't know that there's prominent sleep derangement. OCD, not sure, wondering if people are here. I'm wondering about eating disorders. Just
I see. Okay.
This notion about what? Yeah, sort of. Terrible sleep. Okay, so then forget it. Anyway, what I was wondering is that maybe some of the disorders that involve more basal ganglia versus other parts of anyway. So
it's possible. It's possible.
All right.
No.
So this is my question. So, it seems to me that there are kind of two potential models here. I'm not sure they're completely separable, but one is that you've identified a set of highly pleiotropic genes that generally regulate kind of overall efficiency of neuronal function, and that sleep is one of many readouts that's showing that that's happening. And then the other is that there's some pathway in which actually the behavioral aspects of sleep and associated biological functions are in the kind of causal pathway.
Right.
And so what I'm wondering is, it seemed to me that your early tau experiments, I was wondering whether or not there was a way to use DREADS or maybe optogenetics in order to try to specifically alter the sleep-wake behavior and see whether or not that is what is.
Well, yeah.
Like an impossible, or there may be other ways that you're thinking about sorting that out. Either way, it seems to me incredible.
I was considering, I feel like I have to focus on sleep, right? Because there's just so many interesting questions. I was intrigued, thinking maybe I should dig a little bit deeper, but the funding didn't come, so I stopped. But we do have a little bit more data, like if we activate the neuron there, it affects the REM sleep, and then directly linked to the reduced tau accumulation. We have a little bit data there, but I was thinking to, but it will be very interesting. But we have other data that not related to tau to show that actually ADRB1 mutant neurons are more active, right? So it would suggest indirectly that because ADRB1 mutant neuron is more active, so if you activate this neuron, it can then prevent the tau spreading, right? So that's similar, but not the same with using DREADS to activate it. We did do a little bit of DREADS. I didn't show it today, so.
I guess my question is asking about the mirror side of this. So you talked about the short sleepers. Do you see the same thing, the opposite on the long sleep side?
We just got the first long sleep gene, and our mouse model is almost ready. So, but the gene, I don't want to say it's not published, but it makes sense. It falls the genes feature. It makes sense with the whole picture. It makes sense in a sense that its sleep efficiency is the key, right? So the long sleeper probably is good. Just need more time to get all this function complete, right? It makes sense in that way. It makes sense, yeah.
So Dr. Fu, there's also a couple questions on the Zoom.
Oh, okay.
That I'll ask real quick. So one of the questions from Dr. Ann Glowinski says, "Good morning, Dr. Fu. How longitudinally stable are these sleep phenotypes you describe, and also at what average age do they stabilize?" Do you want to take it?
You mean how? Where is this?
This one right here.
Okay. How longitudinal? I mean, they are stable. I think that. So what happened? I think Andrew maybe know better than this. So some people very early on, like I don't know, 5, 6, 7, 8 years old, already very stable, and some people have to wait until like a teenage year, but at some point, so it depends. I think it varies, and then after they become that way, it's like lifelong short sleeper. Yeah.
Not uncommon for these short sleepers to tell the story that they pretended to be asleep and would read under the covers.
Yeah. Yeah. Yeah.
They had trouble with sleep bedtime.
Yeah. Some can be very early, but some it's like early adulthood. Is there another question? Oh.
Thank you. This is all very exciting. So I have 2 questions. The first is, as an epidemiologist, I just can't stop thinking how these rare mutations or like, these short sleepers, how they can be translated into more of a population-based sleep needs. How can we find, our individual sleep needs that's obviously different for everyone, and not all of us would be like short or long sleepers. And then the second question is, what's kind of the main micro sleep structure that's driving, behind this? What do you think? Is it REM, non-REM, or any spindles that's making a difference?
Yeah. The first question, I don't know how do we use our mutation into find the wide population, but population. But people ask me how do they find their sleep schedule. I always say take a long vacation and don't drink coffee or tea or anything, right? And then figure out what is your best time to go to bed and get up and how much sleep. I think for most of us, right, it's very hard to tease apart all the factors that we have exposed ourselves to. So if you want to figure out, you have to kind of set a month to go on vacation or 2 weeks, right? And the second question that, oh yeah, that's a very good question. So and very important, and we're trying to address that. We don't have an answer yet, but I think that hopefully the problem is to score this is so labor intensive, and we now is very close to have a way to do this by computer, machine learning, and then we can really look into the microstructure and compare short sleep with regular. Yeah, very important. Yes.
Okay. And one last question from Zoom. It says, "Are you looking at sleep apnea as a variable?"
No. We're not looking at sleep problem yet. I think if we understand regular sleep, how regular sleep works, someday we will be able to help with sleep problems, right? And right now we're just trying to understand how normal sleep is regulated. So.
Thank you.
Okay, thank you.